Okay, so thanks, Simon, for joining us today. The stage is yours. We're looking forward to your talk.
All right, thanks for the kind introduction, which made me feel very old all of a sudden. So it's a pleasure to be here, and it's a pleasure to see some old friends here in the audience. Yeah, so I'm gonna get right to it. So I will talk about some recent studies that we've done in my lab. Most of these are not published, and they use a combination of human single unit recordings and computational modeling, and also some behavioral methods to essentially get at the question is how memories are formed and how the human hippocampus manages to form these memory. One of the classical ideas that I think the classical, arguably one of the most fundamental principles in neuroscience is the idea that neurons that fire together, wire together. That was proposed some 70 years ago, over 70 years ago by Donald Hebb. And there is, of course, a very rich literature in the animal field, so using animal models that have shown that these principles indeed hold. But if one looks into what do we know as to whether this fire together, wire together idea also holds in the human brain when humans form episodic memories, I found it astounding as how little we actually know about whether that actually applies, whether neurons actually really synchronize their firing in narrow time windows in order to lay down traces of an episodic memory. And that is the very question that I would like to shed a little bit of light on in the next few minutes. So one synaptic plasticity mechanism that that features very strongly in Hebbian learning is called Spiketimeindependent plasticity. And in its essence, what this principle says is that whether you get long-term potentiation or whether you get long-term depression depends on the sequence of firing. So if an upstream neuron fires quickly or shortly before a downstream neuron responds to that input and fires, then you get a very strong long-term potentiation, right? And if the sequence is reversed, then you get very strong long-term depression. And the important bit here is that the strengthening or weakening, so the synaptic modification here depends on the time windows, right? And it doesn't decrease linearly, the strengthening decreases exponentially. So that really puts emphasis on the very fine-grained temporal coordination of these spikes. And that puts gamma oscillations, I think, right at center stage, because gamma oscillations of the frequency, the very fast frequency, they are one very prominent candidate mechanism to impose such fine-grained neural synchronization. And indeed, we have shown some in a study that was published 2 years ago that the presence of fast gamma oscillations, so gamma oscillations at about 60 to 70 hertz in the human hippocampus does predict successful memory formation. Interestingly, in the same study, we also found a slow gamma oscillation, which is depressed during successful memory encoding, but dominates during retrieval, which links very well with Laura Colgan's very inspirational work, showing that there might be two different gamma rhythms which dictate encoding or retrieval. However, what I want you to keep in mind is that fast gamma oscillations during encoding, we can measure them in the human hippocampus, and indeed they seem to correlate with successful memory formation, which lines well up with the idea that they impose narrow time windows for synchronous firing. Another mechanism that I would like to focus on that features prominently in the literature is theta phase-dependent plasticity, which I think originally goes back to work by Pavlidis in 1988, but the Huerta and Lisman paper I think is a very prominent paper here that shows this mechanism very elegantly. And what they have shown is that if you stimulate neurons in the theta rhythm, then you get opposing changes in synaptic weights, depending on whether you stimulate at the peak or whether you stimulate at the trough. And please note that one stimulus alone is not really enough to drive these changes. It needs a burst of stimuli, which then makes sense to think about these bursts as little gamma oscillations being nested in these theta peaks or theta troughs. So that was where we started off with our first human single unit study, where we basically asked the question, can we see these theta and gamma oscillations in the human hippocampus, and can we see that these oscillations really induce co-firing of neurons when memory episodes are successfully encoded? That work has been done in collaboration with the Epilepsy Center in Erlangen, Professor Hammer, and Matt Self in Amsterdam, and Ramesh Chalvaraja in Birmingham. We managed to record data from about 44 sessions. The data set now includes many more subjects, but the data set that I'll talk about that we analyzed then was 9 patients. It's 44 sessions, so it's lots of data indeed. And we record the single units with these microwires, and from these same contacts, we can also record the local field potential. So you see the raw data here, and you see some spikes riding on the raw data here, and you can nicely split the firing of the neurons from the local field potential by applying some filtering. And the task that we used was this associative episodic memory task, where we first presented the picture of an animal. It was always the picture of an animal here at time 0. Then the pictures of face and the place, or 2 faces or 2 places, appeared 2 seconds afterwards. And then the patient was instructed to make a story that would link all these 3 images together. And the purpose of this task was to get the patient to basically form little episodes, right? The associative rich episodes that would link the seagull here with Brad Pitt and the Taj Mahal. For instance, Brad Pitt goes to India on a holiday trip, and then a seagull comes by and sits on his head or something like that, messes up his hair or something. And then the next trial would start. And after a distractor, we presented the picture of the animal again, and then the patient had to indicate how many images do you remember, 0, 1, or 2. And then they could select the appropriate images that went together with the animal during encoding. And that's the task. And all data that I'm going to show you will be from this time period, basically when the stimuli came on, right? Because this is the earliest possible time window where I think the memory formation process could start, because only then the patient is given the full information, and memory encoding processes would basically any neural processes at this time point would set the stage for laying down the neural traces of a memory. So the first analysis we did in this data set was spike field coupling. So we looked as to whether the spike of a neuron would be coupled to the local field potential. And this can be done in 2 separate ways, depending on the spatial configuration of the spike providing channel and the local field providing channel. So you can think about the combination being local, such that essentially you take the local field potential from the same channel or an adjacent, very closely adjacent channel to where you record the neuron from. And that would mean that essentially the firing of the neuron would be locally entrained to its neighbors. But the coupling can also occur at the distal level, where the firing of a neuron in one region would cause a postsynaptic current in another region. And this is typically taken as a directed measure of connectivity, because indeed there needs to be a synaptic connection in this direction if the firing of one spike leads to a postsynaptic current change in another region. And we first analyzed gamma frequency range between 40 and 80 hertz. And the results show that there was a very clear peak in the fast gamma frequency, very similar to what we've found before, between 60 and 65 hertz. We did not really find a distal coupling. So the gamma spike field coupling, gamma oscillations were in our data set very much a local phenomenon. But the important question is of course, is there a difference depending on memory? And the answer is yes, there is. There is a difference. And this difference takes looks such that during hit trials, so during successfully formed memories, the coupling occurred at the slightly faster gamma frequency, and during mistrials, the coupling occurred at the slightly slower frequency. And this is just one example from one unit. But this result held also at the population level, where across the population, we found very consistently faster gamma frequencies for successfully remembered trials and slower gamma frequencies for not successful, or for not remembered trials. So that was the result with respect to gamma. Now we did the same analysis with slower frequencies, including the theta frequency range. And there we also looked at local and distal coupling. And here the distal coupling proved to be more interesting. So it's important, we found both local and distal coupling in the theta frequency range, but it was only the distal coupling which would correlate with memory. And it correlated with memory just the same way as gamma did, interestingly. We found faster theta frequencies for later remembered trials and slower theta frequencies for later not remembered trials. And this again came out very, very consistently at the population level. So if you take it together, gamma and frequency, gamma and theta, they do correlate the same way with memory, such that the fast frequency would predict a good memory outcome. But their coupling was slightly different, such that gamma was very much a local phenomenon, whereas the theta was a more distal phenomenon that would basically link neurons between regions with each other. You might now ask, was the gamma oscillation also nested in the theta oscillation? And the answer is yes, it was. I'm not going to show you the result for time reasons. If you're interested in seeing those, I'm happy to show them in the discussions. But the point here being is, you know, what's the point of all these oscillations, oscillatory coupling? The point of these oscillatory coupling would be, of course, that they would allow two neurons to engage in efficient communication, right? Such that when the upstream neuron fires, it elicits a theta oscillation in another region that then drives the gamma to be nested into that theta, and that would allow this neuron to efficiently respond to the spike that it's been sent from an upstream neuron. And so we looked at all our pairs that were coupled in this theta gamma firing regime, and looked at the co-firing. That is, when does the downstream neuron fire in response to an upstream spike? And the idea would be that during successful memory formation, that spike should occur earlier than during unsuccessful memory formation, because it is this narrow time window synchronization that matters, right? Fire together, wire together. And here is one example neuron pair where we have one neuron in the left hippocampus that is coupled to the entorhinal cortex, also in the left. And you can see that during successful memory, indeed, the downstream neuron responds way earlier, here at about 30 milliseconds, than during an unsuccessful trial, shown here in red. And this is, of course, just one pair, but this result also held at the population level, such that earlier co-firings did indeed correlate with a successful memory. So that links very nicely with the idea that theta and gamma oscillations synchronize neurons, and you speed up the frequency in both, and you get a more tighter coupling between those, and more tighter coupling would then also, of course, result in more spike time independent plasticity. I would like to say that the oscillations are causally responsive for this change. But of course, we can't do that here because we simply record these neurons and we observe. So all we can do is correlate, but we can't do any causal manipulations. However, what we can do is we can construct a computational model that takes all of our findings into account and drive these neuron pairs with slow and fast theta and gamma oscillations and see what that does to spike time independent plasticity. So in this model, we have full control over all parameters, so we can run a causal manipulation in silicio, if you will. And so here is a successful memory trial simulated in this model with the fast theta and the fast gamma frequency. And we have an unsuccessful trial with the slow theta and the slow gamma frequency, and the gamma is nested into the theta for the successful memory trial and not nested in the unsuccessful memory trial. And you can see already the tighter packaging of these spikes for the faster frequencies. And indeed, when we look at what does this change in frequency do to spike time independent or to synaptic plasticity, as implemented by STDP, we can see that there is indeed a very dramatic difference, such that for the fast frequencies, the hits increase or neurons for hit trials increase their weight within just a time course of 1 second, right? Up to 250%, whereas for misses, it barely goes above baseline. And this model also nicely simulates the observed spike coincidences, where we would show earlier coincidences for hits and later coincidences for misses. So the observed differences in theta and gamma frequencies drive these latency differences in co-firing in this model, right? And they dramatically affect synaptic plasticity. So conclusions. Neurons are coupled to theta and gamma oscillations in the human MTL, and these oscillations slightly speed up, right? And that speed up is related to positive memory outcome. And this speed up of the frequency, this acceleration of theta and gamma frequencies also correlates with co-firing at shorter latencies, which is, of course, consistent with the fire together, wire together idea, and indeed consistent with the spike time independent plasticity. So the next question then that we turn to in analyzing this data set is, what do these hippocampal neurons actually code for, right? So what is the information that these neurons harbor? And there has been one very influential idea that I think guides a lot our ideas of human episodic memory, and that's the hippocampal indexing idea. It was put forward by Tyler and Deschena in the mid 80s, but I think it actually goes back to David Marr, like most or lots of neuroscience does. And you can see new riffs on this classic theme also in Bouchaki's recent book. And the idea of the indexing theory is basically that you have a division of labor whereby the cortex would code all the content, right? But the hippocampus actually doesn't store the content. It simply just assigns an arbitrary index to whatever is going on to the cortex, right? So you imagine you experience your episode, and lots of stuff goes on in the cortex, and basically you just take a random cell in the hippocampus and just assign it to, or a random bunch of cells rather, assign it to whatever is going on in the cortex. So there is no direct link, right? This link is made arbitrarily and it's made on the fly. And then later on, if you try to retrieve this memory, then you would be presented with a partial cue that projects to this index, and then that reinstates the pattern, which is known as a process called pattern completion. So we were looking for such cells that would fulfill this characteristic, that would, in particular, in our experiment where we show these combinations of animals and faces and places, that we were looking for cells that would code for one particular episode, right? That would not code for whether this is a face or whether this is a place or whether that is a particular animal, but looking for cells that would code simply for this particular episode, right? One or two episodes, but not more. And that is work that has been done by Luca Colibius, a PhD student in my lab, who has done really outstanding work on this project. And the answer is, yes, we found cells that behave like such. And here I show you one cell. So that's the wave shape of this single unit. And what you see in the raster plot is on the y-axis, you see the trials. So here we presented some 34 trials. And you can see in the raster plot the firing of that particular cell during encoding shown in purple and during retrieval shown in orange. And then we basically combine the firing rates for both encoding and retrieval by multiplying them. And then we z-score them. And what this gives you is a combined estimate of the firing rate of this neuron. What this essentially shows is that this neuron is basically only interested in two trials. It fires in that trial where the combined firing rate exceeds a threshold, which we placed at 2, and it fires in that trial. Here it fires much stronger. So this particular neuron seems to code for these two trials. And you can see that the cues that we used are very different animals. So one is a bull and the one is a sea animal. So it's very difficult to imagine that this neuron would respond to the animal of some sorts. And another constraint that we put here is we made sure that one trial is associated with two faces and the other trial is associated with two places. So we can exclude the possibility that this neuron codes for some simple content, or indeed that this neuron is a concept neuron, as they have been described by Rodrigo Quiroga beautifully. So we think this is a different neuron. This is a neuron that codes for one particular episode in a very arbitrary way. And we did find more of these units than would be expected by chance. So that's what we called index units. And we defined them in two ways, a very conservative way where we would call an index neuron only an index neuron if and only if it will code for two trials, where one would be a face-face trial, the other one would be a place-place trial, which of course is extremely conservative. We found 5 of those, and that might not seem impressive. However, it is way more than you would be expected to find by chance. When we use a more lenient threshold, where we don't have this constraint that it has to be one face-face, it has to be one place-place trial, we find many more neurons, and again, more than would be expected by chance. And the firing rate of these neurons also looks very, very interesting. So this is the firing rate of these index neurons during encoding and during retrieval. And you can see that during encoding, actually nothing much happens until the stimuli come on, right? Until both face-face or face-place or face-place images are shown. And then the firing rate of these units really goes up. Whereas during retrieval, we see that the units start to increase the firing rate already when we present the cue, which is the animal. And remember that during retrieval, there is no showing of the actual stimuli on the screen up until the patient presses how many images they remember. So this increase in firing rate cannot be driven by the presentation of some images. So what I've tried to show you here is that I think we found neurons in the hippocampus which seems to code for specific episodes. And the firing rate patterns of these neurons cannot be simply explained by simple content coding. So these neurons appear very different from the concept neurons that Rodrigo described so beautifully. Okay, so that's essentially the human single unit work. Now I would like to use the last few minutes to talk about how can we test these assumptions of theta, phase plasticity, or synaptic plasticity, and spike time plasticity in humans without necessarily sticking electrodes in there. In this, to this end, we used a combination of computational modeling and behavioral methods. So this goes back to studies that Andrew Clouter, when he was in my lab, he's now a lecturer at Trent University in Nottingham, and Dan Ying, who is still a postdoc in my lab, did a couple of years ago, where we would present movies that were coupled with sounds, and we would modulate the movies such that they would flicker at the theta frequency range. And we also modulate the amplitude of the sound such that the amplitude would also change in the theta rate. And thereby, we can play around. So this allows us to play around with the synchronization between the video, so the visual input, and the auditory input. We can put them in phase such that they would be synchronous to the brain, or we can put them successively out of phase such that they would be asynchronous. And the data that showed in both experiments, this is from 2 different experiments, showed that memory performance is indeed higher when we synchronize the video with the sound. And as we progressively desynchronize the 2 input streams, memory performance gets worse. And so one thing so when I first saw this result, I was happy, but I was also a little bit surprised, because I was originally expecting this 270-degree phase lag data point to go up again, right? You would expect that theta phase, you would expect it to look like a sine wave. So it should be high for 0, then it should go down to 180, but it should go a little bit up for 270, but actually it didn't. And then back then, I started thinking maybe this is already a first hint for spike time independent plasticity, where indeed you would assume an exponential decay. However, it can't only just be spike time independent plasticity, because we have found this effect specific for theta. So if we flicker at other frequencies involving delta frequencies, much slower, 1.6 hertz, or alpha frequency much higher, we didn't find this difference between synchronous and asynchronous stimuli. So there is both the spike time independent plasticity, so the narrowness of the synchronization, as well as the theta frequency, seemed to play a role, and that was really mind-boggling. And then the pandemic struck, and we were stuck in the lab, not being able to collect more data. So I thought, well, let's just see whether we can model this with a computational model. So that's what we did. Luckily, I happened to find myself a computational modeler in my lab, George Parrish, who is a very, very skilled computational modeler. And Dan Ying, they took on this adventure. So in the model, what we had is a very simple model. We just had basically a bunch of neocortical neurons that we call visual and auditory neurons that we would feed theta waves as we would in the human brain with using the videos and the amplitude modulated sounds. And then these would project to hippocampal neurons, and then the hippocampus would form links between the 2. And we used a theta phase dependent STDP learning rule. So what we basically did is we just used theta phase dependent learning and STDP and chucked them together in such a way that at the theta peak, we would favor spike timing, we would favor long-term depression as guided by spike time independent plasticity, meaning that if the sequence of firing between 2 cells was such that they would indicate long-term depression, then that would be upregulated, right? And if they would occur in such a way that it would lead to long-term potentiation, we would ignore that at the theta peak. At the theta trough, we do the opposite. There we really turn up the long-term potentiation and we ignore the long-term depression, right? As guided by the firing sequence of STDP. And at the middle, we would allow both to occur. So that's our mechanism that we implemented. And to cut the long story short, the data of this model really nicely modeled our behavioral data. Keep in mind that this is two different data sets. For some reason, the students that we collected here had an overall worse memory performance, but you can appreciate that the effect of the phase difference was the same in both data collected by Andrew Clouter and Dan-Ying Wang. And the model nicely followed this trajectory. So it was very well able to simulate the results that we found in the behavior. And the model also successfully simulated the theta specificity of the results. So when we feed the model with different synchronized, asynchronized input streams at the delta or at the alpha frequency, then the model would not produce the difference between synchronous and asynchronous at other frequencies than theta. So to this end, we can say that the results we observed in the humans are consistent with a theta phase-dependent STDP plasticity mechanism. And I think, well, my speculation is that this is what we see in the humans. And to link this back to Huert and Lisman, we also simulated the experiment ran by Huert and Lisman, and our results also nicely simulated the Huert and Lisman results. So this model is essentially able to bridge the gap between, a slice preparation study that was done, in a microscope in a petri dish, essentially, all the way to humans sitting in front of computer screens trying to form memories. So I think that is very useful. So to conclude, synchronizing multisensory stimuli at theta does affect memory. Your memory is better when you synchronize them, and it gradually gets worse as you desynchronize them. The time window for that seems to be indeed very, very short. And theta phase-dependent memory effects can be modeled using a theta phase-dependent STDP learning rule. And this same learning rule is also consistent with the findings of Huerta. Whoops, there's a typo. It should be Huerta and Lisman. All right, so that's it. Thank you for your attention. So the study on the theta gamma oscillations and the single units, that's on bioRxiv. The other studies will hopefully soon appear on bioRxiv. And I would like to thank my lab and my lab alumni and the collaborators who helped with this research, and of course the funding agencies that generously endowed me with the money to be able to do this. And thank you for, for your attention, of course. And I'm happy to answer any questions that you may have.
Thanks, Simon. Thanks for the great talk. I see lots of clapping animations. Okay, so I would suggest that, yeah, to sort of organize the discussion. If you have a question, please put your name in the chat, and then I will ask you to speak up, and then we can hopefully have a good discussion. Okay, there is one question by Abderryan.
So first of all, thank you for the interesting talk. It was really interesting data. So my question about the fast gamma. So based on the work of Laura Colgan, we know that the fast gamma is coming from the entorhinal cortex and the slow is from the E3. So based on your results, the difference between the hits and the misses is in the range of 50 to 70 hertz, which is in animal research is more or less the fast gamma range. So do you think, based on these results, that the entorhinal cortex and CE1 were more involved, or there were different processes happening during these misses and hits?
Yeah, it's a very good question. I think we have two possibilities. that could happen here, or at least two possibilities. One is you have the fast gamma, the entorhinal cortex CA1, and you know that the frequency of that fast gamma is modulated dependent on attention or whatever, you know. And then if the frequency is higher, you know, it's an indicator that patient might be more attended or something, and then memory formation takes place because it allows neurons to fire at the higher synchronization. The other, so in that interpretation, basically we would only see entorhinal cortex CA1 gamma, and the frequency of that fast gamma is, you know, regulated up and down. The other possibility is that you have two generators which are in sort of a, you know, a tug of war and fight for dominance, and that the patient occasionally drifts from encoding to retrieval, you know, even though we record during the encoding phase of the task. Of course, the patient sometimes, you know, retrieves some other stuff that might be not related to the experiment, and that is what's driving the frequency of the gamma. Unfortunately, I cannot see really how we could disentangle between these two possibilities. I think our data is consistent with both.
Another question is about the index units. Do you see any correlation between the index units and what we see in hippocampus with regard to time cells? So do you think this index unit is encoding a specific episode and a specific time?
So yeah, so I think that these index units indeed code for a very specific episode. However, I do not think that these are time cells. We cannot be 100% certain for now, as Luca is analyzing actually, whether these units behave like time cells. But for instance, if we look just at face value at what trials these neurons code for, they don't seem to be neighboring trials, right? So the temporal distance between the trials which these neurons code for seems to not show a clear pattern that would say, you know, these cells, the trials that they code for cluster temporally. Interestingly, and before we got the results for the index neurons, we came up with a computational model that I didn't show you, where we assumed that there is a division of labor between time cells whose job it purely is to index a particular time, and index neurons whose job it purely is to arbitrarily, you know, index patterns in the cortex. And in this model, we've assumed the division of labor and decorrelated both. And our data, I think, is consistent with this model, which, interestingly, we came up, you know, way before we had these results.
Thank you so much.
Okay, then another question by Chung Zhao.
Yeah, thanks for organizing all the things, because I saw it from Twitter, and currently I use Chicago, so it's 9 a.m. for me, and just woke up for an hour. And I like the talk a lot, and I'm a long-term fan of your work. So I was wondering if you have a theta phase analysis on any Sculpt EEG data, because from like following your work for a while, you've done like a lot of Sculpt EEG, like alpha, theta band power analysis. And I was wondering if the phase activities could be reflected on Sculpt as well as the thing that you told in a single unit recording. Thanks a lot.
Yeah. They sure can. So you sure can see these theta fluctuations also using non-invasive methods. One thing we did, and that we will continue to do, is to see, I should say that this work that was led by Maria Wimber and Kaspar Keren, to see whether, if we train pattern classifiers, whether we can link the fluctuation of these pattern classifiers to theta oscillations, right? So, I mean, in the end, if you have a cell in the hippocampus that responds to an input, or you have a cell in the hippocampus whose activity would trigger, the reemergence of a cortical pattern, then you might think, okay, it might not be so different if I record from that particular cell, or if I just record the pattern on the cortex and train a classifier, right, to tell me whether a particular pattern is present. And so that's what we did, or rather Maria and Casper did, and we could show that these patterns fluctuate in a theta rhythm, and you can see these theta phase dynamics as well.
Thanks a lot.
Great. Okay, then Christoph also has a question.
Yes, thank you. Thank you very much. Very nice, interesting work. I wonder about the pharmacological intervention studies that you might also perform, because at least for the animal research, it's relatively well known how this oscillation is caused by the differentiation of NMDA and cholinergic receptors. And the question is, did you ever try to block one of these pathways pharmacologically, or increase it, or what is maybe the role of nicotine, because the muscarinic receptors are some of the crucial oscillatory generators?
Yeah, I wish I would have an answer for that. So so the answer is I don't know. Okay. So we haven't done any causal manipulations, whether it's pharmacology. Well, we started to do some electrical stimulation, but we haven't attempted yet any pharmacological studies. Just to say, that getting the usage of microvirus approved ethically, it's already quite a complex endeavor. Putting pharmacological interventions on top of that will give me considerable,
of course, these are patients. I know, I understand.
However, on the other hand, these patients are, of course, medicated or taken off medication as part of the treatment. So it might actually be, that we could piggyback on what the patient is undergoing as part of the treatment anyways. So that's a good point. That's something I should look into.
Thank you.
Would you know of any epilepsy medications that are used that would have clear effects on the cholinergic system or NMDA receptors?
If I know,
yeah,
these things, not exactly. Well, there are a couple that are, of course, targeting the glutamatergic system. I'm not sure about acetylcholine, but maybe Nikolai knows more about that.
Yeah.
Okay. Are there any more questions? If not, then I would like to thank you again for presenting today, and thank everyone for attending. Yeah, there will be more talks coming up in the next month, so maybe we'll see some of you again. Yeah, as I said in the beginning, I would like to ask the IGSN students that would like to have a certificate of attendance to stay in the Zoom meeting just for like another minute or two, so I can sort of wait till everyone else leaves and then write down your names. And yeah, to everyone else, have a good rest of the day, or a good start in the day, wherever, depending on where you are right now. And thanks again to you, Simon.
Yeah, hey, thanks. It was a pleasure. Just to say that I've blocked out my time until 4:30. So that for you, that would be 5:30, I suppose. So if you have any questions or anything, just drop me an email and I send you a Zoom link and we can talk it out.
Great. Thank you.
All right. Bye, everyone.
Bye.
Thank you. Bye.
Bye.